I know the word.
Really? What is that?
What is that?
Yeah.
And
Not a text message, is it?
sorry, I didn't get to be on my phone.
No, I knew you were right there.
Oh.
Good, thank you. How are your days going today?
I'm picking up turkey at 5:15.
Like what time?
Taking it like a remote.
And I'll be on this road, wherever the one right here. At like probably 5.
Have you, in the isolated room?
Yeah, I'm gonna go a little study.
Oh, okay.
I'm gonna be busy.
I've taken like,
Yeah.
They wanna see everything.
Yeah, I've done my
Okay.
I can't scratch me for you, I have to use a white.
No, I've never done that. I've never done anything like
Yes, I
that.
should show you. Just so you know.
Well, good luck with that, yeah. How's it going, Blessed? No. How you actually had everything for that.
Thank you.
Good, how are you?
Good. When you get a chance, can you email me that picture?
Those photos, oh yeah. Sure, okay.
Because you're the only one that got positive results.
Oh, really?
I want to show everyone.
Okay, yeah, I'll do that.
How are you doing today?
Good, how are you?
Good, excited about that. Next week,
Yeah,
you're leaving a little early.
We were just talking about that with our team. We leave on Thursday and tomorrow waiting for the sun and nice weather. It's nice out today, so
Where you going?
like,
Where you going?
we're both in Florida.
Oh, nice.
Yeah. It'll be nice.
Did you go last year as well?
No, last year we went to Kentucky.
Oh, okay.
Yeah, so we have a nicer spring break this year.
Yeah. Very good. How are you two doing today?
Yeah.
Good. Are you looking forward to spring break?
Yeah. I know the website with you, but I'll be on the website.
Pretty good. I always like break.
Yeah.
Brady, how much you're like studying during spring break? Studying more time for studying?
I won't be here Friday. We have a playoff game.
Okay. Well, good luck with that. So is it home or away?
That's in
Indianapolis or something?
It's more, it's like Hammond.
I'm excited.
It's like 4 hours away.
Good luck with that. Well, good afternoon, everyone. What is it, February 18th? Is that right? I know it's kind of blur after a while, so yes, it's February 18th, it's Wednesday, and I'm going to lecture for about 35 minutes here, and then we're going to run back to the lab and check on your bacteria that you put in the thioglycolate tube, because some of you actually have growth, some of you don't, but that's okay. We're going to go check it out. So that's the plan for today, 35 minutes, try and stay awake for 35 minutes, and then we're going to go to the lab. All right, today we're starting chapter number 11. So just as a friendly reminder, don't forget to upload your chapter notes on the blackboard. So you just finished chapter 10. So, again, no rush or anything, but we do have your next exam scheduled for next week on Friday. So next week on Friday, but if you're not here on Friday, I will have the exam ready earlier than that. So just let me know so we can make arrangements for that. All right, chapter 11, what is it about? We're looking at the diversity of bacteria and archaea. So we're looking at a lot of different kinds of bacteria in this chapter. I don't know if you remember, but at one point in this course, we talked about how new species are being discovered every year. Do you remember that number? Roughly, something like 16,000 new species are discovered every year. And that spans all of the domains. But that also includes the prokaryotes, the bacteria and the archaea. A lot of the 16,000 new species fall into this kind of a category here. So, we talked about some of the reasons for that. There are organisms that inhabit areas that are, very difficult for humans to be at, like at the bottom of oceans, for example. So the idea is that, there's probably, even more prokaryotes out there that we're not even aware of. Now this chapter here, we're on chapter 11, focuses on these three main areas here. So we're looking at diversity, the variations that we see in metabolism. So what do they use for energy, for example? Do they use oxygen or not? We're also going to be looking at differences in where they live. So that's their ecology. So some of these organisms prefer to live on land. We call that terrestrial. Some prefer the ocean or lakes. That's the aquatic environment. And then we're going to see that there's a number of medically important microbes or pathogens, and they have preferences for where they prefer to grow, either in or on your body. Okay. So we're going to look at some examples of those. So, this chapter has a lot of examples. I'm going to pick some out for you. There's it's really too many to really master them all, right? But that's why we have Bergey's manual. I mean, remember Bergey's manual lists everything that's known. Like, we don't need to know everything. But nevertheless, it's nice to have some knowledge of what does exist on the planet. So we're going to start off with this slide that looks at again, we're looking at variation in metabolism. We're starting off with anaerobic chemotrophs. So let's look at that first sentence there. It's believed that the planet Earth was anoxic for about 1.5 billion years. Okay, anyone know what we mean by anoxic?
No oxygen.
No oxygen. So try to imagine a time, not last week, but a time when there was no oxygen on the planet. I can't even fathom that. I don't know about you, but scientists believe that that there was a time when there was no oxygen, and yet there was life on the planet. So that's something that's like a foreign concept to us. And that original life was prokaryotic, and it may or may not have developed spontaneously on this planet, or as I mentioned before, it may have come from outside of our planet, like from Mars. So anyways, originally, think of these early primitive prokaryotic organisms as being anaerobic, performing anaerobic respiration. So what do we mean by anaerobic? In the absence of oxygen. Now I know that may be confusing when you pair it with the word respiration, because a lot of times when we're thinking of respiration, we're thinking about breathing in oxygen. But we need to think about cellular respiration that can take place in the absence of oxygen. So that actually does occur in our bodies. So we'll look at that in a moment here. So even though the world today has ample amounts of oxygen, there are places where there is very little or no oxygen. And so this slide here at the very bottom, describes, like where you can find some of those places. For example, in our own earth, in the ground beneath our feet. In the if it's flooded with water, that will remove air from the pockets that are in soil, causing them to be tightly compacted and oxygen deprived. So even though we have all this air with oxygen, right underneath our feet can be an area where there's oxygen depleted. Same idea in large bodies of water. At the surface, there's a lot of aeration going on of water. But as you travel farther down in the depths, there is less and less. And interestingly, even your own body, even though you breathe in, you inhale lots of oxygen, in your intestines, there's actually very little oxygen there. And that kind of makes sense. Like when you're consuming foods, you're not, hopefully you're not swallowing air. you're consuming solid foods and liquids. You're not supposed to be inhaling all this air when you eat. So as a result, your intestines are getting oxygen deprived as well, and you have microorganisms in there that actually are thriving in that anaerobic environment. All right, so we learned about these anaerobic chemotrophs, and now we have a slide about the chemolithotrophs. So we're talking about organisms that use inorganic chemicals for energy. So inorganic chemicals here. So here's a nice example of that. The source of energy here is hydrogen gas, H2 here. And that's a source of energy because of the electrons that are present. Yes, electrons can carry energy. I mean, that's the basis of electricity that we have. We have electrons flowing through copper wires above our heads. That's carrying energy. So, when you look at hydrogen, I know you only see some H's there, but you're maybe wondering, where are the electrons? All I see is a big H. Well, if you remember from chemistry, hydrogen is composed of one electron, one proton. Okay? So, wherever you see a big H, wherever you see a hydrogen, you're also looking at an electron. Okay, and then we have this process where something gains electrons, something loses electrons. Do you remember the term for that? Gaining and losing electrons? Oxidation and reduction. Excellent, excellent. So those two go hand in hand. When something loses an electron, it doesn't disappear. It's accepted by something else. When we have, what do we call that? Conservation of matter in the universe. Okay, so the idea is that the hydrogen is the source of electrons, and then when you see this kind of a notation here, this represents a reaction. So you remember what you call the components at the beginning of a reaction? Like, what do you call all this stuff on the left of the arrow? What's the general term for that?
Reactants.
Reactants, excellent. And then what do you call the stuff that you end up with? Products, that's right, there you go. So you have electrons here, and then the idea is that they're being passed on to CO2, to that carbon there, and as a result, when you look at the products, you end up with methane. So organisms that release methane as a result of this reaction here are called methanogens. So methanogens release methane. Now, methane is actually the main component in natural gas. So like when you turn on your gas lines in the lab, you know, most of that is methane. But what you're smelling is actually not necessarily methane. There's actually an additive added to natural gas that'll kind of give it a distinct odor to let you know if you have a natural gas leak. But the more truer smell of methane is what you notice from sewage. You know, the stinky stuff from like manure. Okay, that's methane. Okay, so remember that these are being generated in an oxygen-deprived environment. So like from intestinal tracts. Now look at this interesting statistic here about cows or cattle. Would you like to have been the person to calculate how much methane is released from an average cow per day? Wouldn't that be a fun job? Somebody had to do it. But look at this number here. 10 cubic feet per day. So you guys know what the length of a foot is, right? So multiply that by 10 feet. That's longer than you are high. 10 feet long, 10 feet tall, 10 feet deep. And there's your 10 cubic feet of methane from one cow, one day. But you know, there's more than one cow on the planet, right? There's lots of cattle. You know that there's more than one day a year, right? So if you do the math, there's a substantial amount of methane that's generated from livestock. And why is this a concern? About methane. What's the problem with having all this methane in the atmosphere?
Does it also contribute to global warming, like the greenhouse gases?
That's right. Methane is a greenhouse gas. Okay, it's a greenhouse gas. So I know people kind of compare these gases to like the glass panes in a glass greenhouse. They allow radiant heat energy in, but not out. Okay. So it's unfortunate, but you know, methane production is actually contributing to what we call global warming and climate change. So, so there you have it. I know, I'm the bearer of bad news, right? All right, so you got anaerobic chemolithotrophs, but this slide gives us information about the anaerobic organotropes, the chemoorganotropes, those that use organic chemicals for energy. Now, it's still in the absence of oxygen, but yet they're able to carry out respiration. So the whole purpose of respiration is to generate energy. Okay, like ATP energy, that's the whole idea behind energy conversion here. Okay, so let's look at an example here. We have a bacterium called Desulfovibrio. Desulfovibrio. The first part is named because it uses sulfur or sulfate. So in this diagram down below, we see sulfur, and it generates, based on this oxidation reduction reaction, it's generating hydrogen sulfide over here on the far right. So that's a gas. So again, they're using organic compounds as their energy source, but they're releasing hydrogen sulfide as a result. So what does this smell like? Anyone? Sulfur. What does sulfur smell like? Yeah, that's exactly right. Yeah, like what they call rotten egg smell. not real pleasant, right? So every once in a while, when you're traveling around the world, you'll encounter an area like that, and you'll know, oh, we got some sulfur-reducing bacteria here. All right, and then some of those anaerobic chemoorganotropes are capable of more traditional fermentation. So, for example, on this slide here, we have glucose as our energy source being converted into pyruvate. Okay, we know this reaction, this part, by a name. Anyone know the name of that process? Glucose being converted into pyruvate.
Distillation.
It's part of
Okay, so
it's like the beginning part.
Yeah, it's
It's actually before that. Anyone remember the beginning steps of cellular respiration? You take glucose, you're splitting it in half to generate 2, 3 carbon compounds. The 3 carbon compounds are called pyruvate. Glycolysis! You nailed it there. This is glycolysis. This is a reaction you perform, and then the idea is, if you have sufficient oxygen, then you're going into aerobic cellular respiration. But in the absence of oxygen, create lactic acid, also known as lactate. So you guys perform this one too. So think about under what conditions do you perform cellular respiration when you don't get enough oxygen? Think about a time in your life when you are generating energy from the food that you eat, but you're not getting enough oxygen. When you're running, exactly right. When you're exerting yourself from exercise, you feel like you're out of breath because you're not getting enough oxygen. So under those circumstances, you need more ATP energy, and you have lactic acid fermentation kicking in. So you perform it, but so do bacteria. They perform this reaction too. Okay. Like what? Well, for example, like Clostridium here. Clostridium. So that's the name of a genus. And they're known because they produce endospores. So, you know, we did the test, the chemical stain yesterday. And as it turned out, no one had a bacterium that produced endospores. So, on one hand, I was disappointed because I kind of wanted to see that, but on the other hand, what are we dealing with if we do have endospores? So if we do have endospores, we would have destroyed that bacteria because we're dealing with some serious diseases at that point. And like these are normal soil inhabitants. So, but the idea is that if we're growing them in the laboratory, now we're growing millions of them that could cause some serious problems. Let's look at this first example. The C is for Clostridium, and then we have tetani, Clostridium tetani, which is the causal agent of a disease called tetanus. So this one is interesting because it makes a toxin. So Clostridium tetani makes a tetani toxin, and this toxin causes like severe muscle contraction. Well, that doesn't sound so bad because you contract, relax your muscles all the time, right? But you do that voluntarily. This is involuntary muscle contraction. So it's like against your own will, your muscles contract. And I've actually seen some like sketches. This is like in the days before photography of people who had severe contractions of their back muscles. Not forward, but they're contracting backwards. Backwards to such an extent that they actually break their spines. That's how bad it is. Okay. So we don't want that. Okay, some more anaerobic bacteria here. We have Streptococcus. Now, Streptococcus is capable of both anaerobic and aerobic growth. So that's what we call a facultative anaerobe. So it actually prefers oxygen, but can grow in the absence of oxygen. And in about 10 minutes here, we're going to run down to the lab, and we're actually going to see that some of you have these facultative anaerobes growing in your test tubes because you can see growth at the top of the tube where there's plenty of oxygen, and growth at the bottom of the tube where it's oxygen depleted. Okay, so some of you may have Streptococcus. So let's see here. So you remember that metabolic pathway that showed glycolysis, glucose to pyruvate, pyruvate to lactic acid. So that's similar kind of pathways operating in Streptococcus, and there's a lot of different ones. Some are associated with disease, but here's a fun one. One of them is actually used to make yogurt. Streptococcus thermophilus. So you have the acid that's used to denature milk proteins. So it causes them to curdle, solidify, and it makes yogurt. I did notice in the lab, like there's a prep room in the back, and there were some things that were labeled, for yogurt making only. So, and I've actually had students make yogurt before, but unfortunately, they all got diarrhea afterwards. So after that, I stopped making the yogurt. I mean, the idea is that it's a shock to the system, right? When you put all that bacteria into your body can't handle it. It's not used to it. Okay, here's another one of those anaerobic bacteria, Propionani bacteria. Propionani. So this was a fun one because it, through this fermentation process, it creates carbon dioxide. So that's the same as like when you're using baker's yeast to, produce bread. The air pockets in bread are produced because of the carbon dioxide that's released. Similar kind of concept is happening in this case with cheese making. So after the pockets are produced, the cheese, the milk proteins solidify, and you'll end up with these small little holes, and we call that Swiss cheese. So anyone like Swiss cheese? Okay, yeah, it's pretty good, right? I like it too. And it was produced with the help of these Propionani bacteria. Okay, what does it mean to be an obligate anaerobe? Anyone? Obligate anaerobe? What? Obligate anaerobe. I don't know if I misspoke there. Obligate anaerobe.
Only works in non-oxygenated places.
Exactly right. It only grows in the absence of oxygen. Okay. Well, what do you think? Do you think any of your bacteria that you're growing in your petri dishes, are they going to be obligate anaerobes? No, because they're growing in the presence of oxygen, right? So your bacteria will not fall into that category. Okay, and then we have phototrophs. So instead of looking at chemotrophs that use chemicals for energy, now we're looking at bacteria that use sunlight as their source of energy. I know when we think about photosynthesis, we're thinking about plants, how they're generating oxygen for us. But there's actually photosynthesis that can take place in the absence of oxygen. So that's anoxygenic phototrophs. So think back to when there was a time when there wasn't any oxygen in the atmosphere on the planet. So it's that time, under those conditions, where it was believed that these organisms first appeared on the planet. And look at this chemical reaction here. So this is very similar to what you've learned for photosynthesis, right? But instead of hydrogen sulfide over here, what do you normally see in photosynthesis? You see right here, this one? Right. So normally you see what for photosynthesis? You see water, that's right. And then you see oxygen here instead of sulfur. Okay, but then the idea is, well, this is probably the form of photosynthesis that developed first on the planet. Okay, so what the heck is using hydrogen sulfide and producing sulfur? This one. Now this is interesting. It's called chromatium. Chromatium. And isn't that a super cool purple sheen to it? So it's kind of like algae that we see in our local ponds and lakes, but try to imagine, what if that green was a purple? I like it. So this is our purple sulfur bacteria. again, growing using hydrogen sulfide and releasing sulfur. So probably people don't like that, right? Because it smells like rotten eggs, but I sure like the color. Well, this one might be a fun place to stop. I like color. So let's take a moment here. We're going to pack everything up. We're going to go down to the lab, take a look at those thioglycolate tubes. You don't need to wash up. You don't need to put on the lab coats. We're just going to look at the tubes. We're going to make recordings. I'll pass back the lab sheets to you so you can make recordings on it. Okay, so I'm going to shut down and then meet me at the lab.